this lesson today, I'll talk about structure of RNA and its different types. RNA stands for ribonucleic acid. Ribonucleic acid is another type of nucleic acid. You know, there are two types of nucleic acid. DNA, which stands for deoxyribonucleic acid, that we have discussed in another lesson, and RNA, which stands for ribonucleic acid. There are three types of ribonucleic acid involved in protein synthesis, that are mRNA, messenger RNA, tRNA, that stands for transfer RNA, and ribosomal RNA, that stands for, that is represented as R, small r, and RNA. I'll talk about the structure of each RNA one by one. mRNA is a single-stranded RNA molecule. mRNA is synthesized by a process called transcription. If this is a DNA molecule, a DNA molecule has, this is a DNA molecule. A DNA molecule has coding and non-coding regions. Maybe coding regions, and non-coding regions. So this may be non-coding region, and this is a coding region. During transcription, one of the strand acts as a template strand. To see more details, you can watch another lesson on transcription. So this is a template strand. This template strand has coding and non-coding regions on it. During transcription, an mRNA is synthesized, which has two types of regions. One that is copied from non-coding region, and other which is copied from coding region. So you see this region of mRNA, this is an mRNA, stem RNA. So non-coding region has been copied by transcription, so non-coding region is copied into introns, and coding region is copied into exons. So in a premature mRNA, there are alternating exons and introns present along its length. Before this, mRNA comes out of the nucleus, it undergoes mRNA processing. mRNA processing involves one of the processing of mRNA. So one of the process during the maturation of mRNA involves removal of introns and rejoining of exons together. So these introns are removed and these exons are joined together to make a mature mRNA. So this process is called mRNA. This removal of introns are removed. This removal of introns from RNA is known as mRNA splicing. And after removal of introns, a completely coding mRNA is formed for the synthesis of proteins, which is now mature mRNA, and it is ready to come out of the nucleus in eukaryotic cells. Mature mRNA has a complete coding sequence of nucleotides on it, which is copied from DNA by a process called transcription, and it comes out of the nucleus and into the cytoplasm in eukaryotic cells, where the ribosomes bind on it and translate to make proteins. This mRNA has two different terminals. On one end is 5' cap, and on the other end is 3' cap. It has a site for binding of and the start of the protein synthesis, and it is a specific sequence of 3 nucleotides at the beginning, which is known as start codon, where the protein synthesis or polypeptide synthesis begins. And this start codon is always AUG, and it codes for an amino acid known as methionine. Whereas the other end has a specific stop codon. There are 3 possible stop codons. These stop codons include UAA, UAG, and G, A. These three codons do not code for any amino acid, and this is where polypeptide chain is terminated. The region between the start codon and stop codon include the coded sequence of nucleotides or triplets, which is complementary to the coded sequence on the template DNA strand, and it codes for a polypeptide, codes for a specific polypeptide. And this information copied from DNA template is complementary to the coded information on DNA template. If a DNA template has, for instance, this coded information on that you can write, obviously this would have some specific sequence, but you can write randomly any sequence we have already discussed that this coded information is read in a series of 3 consecutive nucleotides together, and this is known as a genetic code or a triplet code, and this code is non-overlapping. Means this each code is specific and is read from one end to the other end in a specific order. When it is copied onto an mRNA molecule by transcription, this mRNA molecule has the complementary nucleotide sequence to the coded sequence on the DNA template. So this is a DNA template. So the first code on a DNA strand is ATG. So this will have a complementary sequence of nucleotides. Against U, A, it has U. Against T, it has an A. And against G, it has C here. So this complementary sequence to the code on DNA molecule is known as a codon. So the sequence of other codons is also complementary to the other codes on this DNA molecule. And so on. And these codons code for a specific amino acid. So the amino acids are joined in a specific order. This first codon codes for first amino acid, which is specific amino acid. We don't need to know the name of an amino acid coded by this codon. Then on a second position, there is another amino acid, a different amino acid. We have another amino acid and another amino acid, and we have another amino acid. These amino acids are joined by an enzyme, peptidyl transferase, which is present in a ribosome, and it makes a long chain of amino acids in a specific order, which makes a polypeptide chain. So this is how polypeptide chain. So this is how an mRNA is involved in protein synthesis. Transfer RNA also has a single-stranded RNA structure like mRNA, but it differs from mRNA in a way that it is folded in such a way that it has three hairpin loop-like structures, making a three-leafed clover shape. One of the hairpin loop has a specific sequence of 3 nucleotides here. So this is a specific sequence of 3 nucleotides here and is known as an anticodon. This anticodon is specific and it has a specific amino acid attached at one end of this RNA molecule. So this is an amino acid attached here. These two strands are held together by hydrogen bonding in between them, and there is a complementary base pairing. If this is an A here, the other will have U here. If this is G here, this will have C here. If this is C here, this will have G here. If this is A here, this will have U here. And these two parts of the same strand, which is folded upon itself, are held together by the hydrogen bonding in between them. So this is hydrogen bonding, hydrogen bonding. So as there are 20 different types of amino acids, so at least there are 20 different types of transfer RNAs which are involved in protein synthesis. So you do not need to know the 20 different types of these transfer RNAs. Each transfer RNA has the same structure, only these transfer RNAs differ at an anticodon and this amino acid present here. This anticodon is complementary to the codon on an mRNA molecule. So this transfer RNA binds with an mRNA on a specific codon which is complementary to the anticodon. Okay, so let's see if I simplify the structure of a tRNA as this is tRNA. This tRNA has an amino acid attached on one end. There's an amino acid attached here at this one end, and it has an anticodon at this end. This anticodon is complementary to the codon on an mRNA. So if a codon on mRNA is UAC, anticodon will be AUG. This anticodon will be the same as that of code on a DNA template, except a DNA molecule or a DNA code will have, this is the code here, which will have thiamine instead of a uracil here. Otherwise, an anticodon is the same as that of a code on DNA, except an anticodon has uracil in it, despite of a thiamine in the DNA code. This AUG anticodon will have tyrosine amino acid attached with it. The other tRNA that attaches at the second position will have an anticodon G, A, and it has an amino acid at the other end attached, leucine. The amino acids are represented with these three letters. You don't need to remember their names, and you also don't need to learn their symbols. However, you should understand that there are amino acids attached with this tRNA, and these are specific, and they have specific amino acid attached at one end, and an anticodon is specific on the other end. At the third position, the transfer RNA that attaches with an mRNA will have an anticodon which will be complementary to ACC, and this will be UGG. This transfer RNA will have an anticodon UGG, and amino acid attached here at the other end will be threonine, and threonine is represented with THR. At the fourth position, this will have, at fourth position, this will have a tRNA attached here, which has an anticodon complementary to UAG, and this will be AUU. This AUU codes for an amino acid isoleucine, which is represented with ILE, isoleucine. At the next position, the tRNA that attaches here has AAU anticodon, and at the other end, the amino acid attached with this transfer RNA will be leucine again. So you see there are two codes possible for the same amino acid leucine. So that is why this genetic code is known as degenerate. This GAA and this AAT, they both code for the same amino acid leucine. So this is how the transfer RNAs will bring these specific amino acids to a ribosome, and ribosome then attaches these amino acids together by peptide linkages in between them, and this makes a polypeptide chain. These transfer RNA, after they have transferred, an amino acid to a growing chain of polypeptide or to a growing chain of amino acids. These are detached from here. They are moved to the cytoplasm and they bind with another specific amino acid and they bring it to the ribosome again. So this transfer RNA will be detached from here and it will be separated from an amino acid and it will bind with a specific amino acid which is present in the cytoplasm here, and it then transfers it to the growing chain at the next position on an mRNA again. Ribosomal RNA is a single-stranded RNA which has been folded, and it is associated with the proteins, and it makes a component of the ribosomes. So there are proteins associated with this. If this is ribosomal RNA, there are proteins associated with it, and this makes a ribosome here. This is smaller subunit of a ribosome, and this is the larger subunit of a ribosome. These two units can be separated, and here it has a slot for a binding of an mRNA into the ribosome. Transfer RNAs bring amino acids to this ribosome, which joins them in a specific order, making a polypeptide chain. This we'll discuss in another lesson in the protein synthesis in detail. Thank you.